Kia ora koutou, ko Andrew Sargent tuku ingoa. I am a teacher at Te Kora, based in Whanganui Atara, which is Wellington. I'm here to welcome our, sort of guest, who is the Head of Science at St Patrick's College in Wellington. That's Doug Walker. And he will be running through the Level 1 Science genetics paper, or genetics tutorial today. Just a little note from our sponsor. These webinars are made possible with the help of Study It. So Study It is an education forum where students can go for help for all different subject areas. And if you look in the video description, you'll see a link to Study It. So while we go through this presentation, if you've got any questions, you just chuck them in the chat, and I'll either interrupt Doug if they're worth sort of interrupting for, or I'll just collate them for the end. Right, so I'll hand over to you, Doug.
Kia ora koutou, and thank you, Andrew. Okay, can I just check that you can see my presentation?
I can see it fine. Yep, it's just popped up now.
Cool, thank you. All right, so this presentation should have been linked for you as well in the chat, I believe, by Andrew. So feel free to open it up and have a look. It's also got the stuff from last year tagged on at the bottom. So there's quite a lot of bits and pieces in there. And as Andrew said, feel free to type questions in chat, and he'll either interrupt me or save them towards the end. So for your genetics exam, which is next Tuesday, there's a few resources that you might not be aware of. Andrew's mentioned Study It already. And there's also No Brain Too Small, which is a brilliant repository for exam questions. A whole load of like revision notes and bits and pieces. I think they've got some flashcards on there for genetics. And the questions are organized by type. So if you know that there's a particular concept that you struggle with, then you can get onto No Brain Too Small and really focus on those ones that you need to be upskilling in. There's past exam papers from NCEA, and if you haven't done those, you really should be getting on with a few of them over the weekend. I always recommend to my students that they do at least 5 past exam papers for every exam they're going to be sitting. And if you've done that, especially in Level 1 Science, there's not going to be any surprises coming your way in the exam. You'll see the same sorts of questions again and again. And I've linked the marking schedules in there as well, because there's no point doing the exams unless you're marking your work and getting an idea of whether you're on the right track or whether you need to learn a little bit more about a concept. So what we're going to cover tonight, a few different bits and pieces. I'm going to go through, for genetics, I've pulled out a whole load of the commonly assessed questions. So looking back at previous years, you can see some really strong patterns in genetics, and some questions that occur every 4 or 5 years, but not as often. So we'll go over those. I've linked in some definitions here as flashcards. These are just ones that I found online. And the definitions for genetics are really important. There's a lot of unusual terminology that you might not have encountered previously in science. And you need to know what those words mean and how to define them. That often forms a foundation of your answer in the exam. So use the flashcards as a sort of way of active recall, trying to memorize both the term and the matching definition. We'll have a look through the examiner's report, and there's some points on there. I haven't updated it for this year, but the points on there are often very similar. And so we'll go through the sorts of things that you've got to make sure you can do before you go into the exam. And then for the most part of the presentation, we'll go through the 2021 paper. I'll run through the questions that are in that, and I've pulled out a couple of other ones from previous years as well, just to reinforce some of the key concepts that you might encounter in the exam. And as I say, as we go through, feel free to ask me to go over any points or to throw other questions my way. And if we've got time, we'll certainly do that. All right, so this little table has got the years of the exams along the top, and those are hyperlinked if you use the presentation yourself. So you can use those to jump to the relevant paper. And down the left-hand side, I've got the concepts. So you can see the sorts of concepts that I've identified as being a question type in the papers, and then the frequency of them. So the little dash in the box or the little slash in the box means that they did occur in that year. So we can see Punnett squares have occurred every year all the way back to 2014. So there's a good chance, I would say, that Punnett squares are going to be in your exam in some shape or form. In 2021, so last year, the Punnett squares were only related to sexual determination, so determining the sex of offspring. But normally it's more along your sort of standard monohybrid cross patterns. You can see that phenotypic ratios and the link between phenotypic, genotypic ratios and chance occur less frequently. So maybe 3 years out of all of those years that we've looked at. If we go to inherited versus environmental traits, however, then we can see that that's come up 6 times over the last 8 years. So it's a bit more of a common one. And you'll notice that I've highlighted in bold the concepts that come up most often. So these are definitely ones that we're going to try and hit tonight and make sure that you're aware of for your exam. So the relationship of terms such as, what is DNA and how does it link to a gene, to alleles, to genotypes, phenotypes, mutation. That's another one that seems to come up every year. Meiosis and fertilization and how that links to variation in a species is a very common one. Variation and survival looks like it's come up every year for the last 8 years. Interestingly, in 2021, that came up twice, which is very unusual. Genotype and phenotype is quite a common one. And pedigree charts. So it didn't appear last year, didn't appear in 2018, have normally appeared. So even though that's not in the 2021 paper, I'm hoping we'll have time to drop into one of those earlier questions and make sure that you have a chance to see how you would interpret the question. Okay, so there's quite a lot on this slide. I'll try and go through it fairly quickly because you can always go back through and read through it more slowly. This is quite negatively phrased in terms of the report. This comes from an examiner's report that is written after every external exam by NCEA. And so you can go into this, it's freely available on their website, and you can see the sorts of things that commonly stop people getting from, say, getting an achieved grade or getting a merit grade. So I've pulled out just the bottom level here, what's stopping people from getting past not achieved into getting the credits. So you've got to be able to show understanding of genetic language. So the terms like homologous, homozygous, genotype, phenotype, all of those weird and wonderful words we get with genetics. Make sure you know the definitions, and that's where those flashcards are going to come in really useful. You've got to be able to identify something as heterozygous, homozygous, based on the combination of alleles. Or conversely, if you're told something is big T or little t, you've got to be able to identify that as homozygous. Fertilization, there's a bit of a misconception often here that fertilization is a process that occurs before or during meiosis. So meiosis creates those sex cells, the sperm and the egg in humans, that then go on to fertilize. And so you've got to be quite clear in your understanding that meiosis is something that happens, often long before fertilization ever occurs. We'll go into that tonight as well. When it comes to variation, you've got to understand that variation is something that happens naturally over generations. It can be a random, initiated by a random event as a mutation, and it's not something that happens in response to environmental change. It's just that the new versions of a gene that can be created through mutations become strengthened in a population as a result of environmental change. So it's not that there's suddenly the climate changes and species rush to adapt to that. That's not the way it works. So you've got to be aware that natural variation exists in any population, and then an environmental change can reinforce certain favorable characteristics within that variation. Sometimes you get asked about what causes genetic variation within a population and what the benefits are. That's one of those questions that comes up very frequently. And so you've got to be able to talk about the causes and benefits of genetic variation. Occasionally, you might have to talk about it might be useful to be able to think about some of the downsides if there's a lack of genetic variation as well. So Punnett square is one of those things that seem to be, a common theme throughout all of the genetics papers. You've got to be able to complete a Punnett square based on the information you're given in the question. And recognize that both plants and animals reproduce sexually. Some plants reproduce asexually as well, but sexual reproduction in plants is very definitely part of nature and can lead to variation within populations. And when you're given resource material such as the images, the wording in the questions, you've got to be able to interact with it, to be able to get information from it, and then to use that information to form your answer. You can't get away with just repeating what's in the question. And pedigree charts, so you've got to be able to make sure you can make sense of a pedigree chart. I've put in some links there. These are ones that I used last year, but the Conceptions About Genetics from Science Learning Hub is a really good link. If you're struggling to understand some of the basics, go and have a read of that, and hopefully some of those things will start making a little bit more sense. So we'll get into some of the exam questions, but before we do, is there anything popped up in chat, Andrew, that you'd like me to address now?
No, there's nothing so far.
Awesome, thank you. All right, so this is the 2021 paper that people would have been sitting about a year ago in your place. John and Nolene have 3 children, 2 girls and 1 boy. Complete a Punnett square showing sex inheritance. Now, sex inheritance doesn't come up an awful lot in the exam, but it's really important that you understand how it works. And this is the old females of a human population have an XX chromosome and males have an XY chromosome. And so in that way, we can pop in the chromosomes for the parents. The XX, again, is the female. The XY is the male. Interestingly, the Y chromosome is shorter than the X chromosome, and that means that certain genes that occur on the sex chromosome, or the X chromosome, don't have a counterpoint on the Y chromosome, and that's why males suffer a much higher proportion of certain genetic disorders if they're located on the X chromosome, because you've only got one shot of having a normal allele, if you like. And if you get an allele that codes for a genetic defect, then you don't have a corresponding allele from from the Y chromosome to counteract that. So male color blindness is a classic example. I think about 9 out of 10 cases of color blindness are in males as opposed to females in the human species. So in the Punnett square, we pop in those possible chromosomes that can populate the gametes, the sex cells. Remember that the gametes are haploid. They contain half of the genetic information of the parent cell. And so that's how we would fill this one out. And then if we go down, we're just filling in whatever chromosome is listed in here, and whatever chromosome is listed in here populates this box. When we come up to this box, we're taking this chromosome, blue X, and this red X to fill out that box. And so we can see from that the expected sex inheritance ratio from this Punnett square is 50% female and 50% male. We get that from there's 2 lots of XX, which would code for female, and there's 2 lots of XY, which would code for male, which is why the majority of the population throughout the world is about 50-50 split male and female. If you actually look at the actual figures, though, females often edge out as a slightly higher percentage across the global population, and that's often put down to slightly more dangerous or reckless behavior on the parts of young males, and a slightly higher death rate in young and adolescent males.
It's just a quick question. Someone asked, does it matter if you put gamete instead of haploid cell?
No, I'd normally say, if I was using like full language, I'd say haploid, gamete, just to demonstrate to the examiner that I know that the gamete has half the genetic information. You can say, you'd probably get away with saying haploid cell or haploid gamete. Haploid gamete's a little bit more specific, I suppose. Good question. So how is sex of children determined in humans? Well, I think I've kind of covered that verbally already, but just to recap, the male has an XY pairing of chromosomes, and so his gametes can contain either an X or a Y in the sperm. The female has two X chromosomes, and so her gametes in the egg can only, I say gametes in the egg, sorry, the egg is the gamete, so her egg cells can only contain a single X chromosome. And that means that you are combining always an X chromosome from mum and an X or a Y chromosome from your father. If you get an X chromosome from dad, then that's going to make your sex female. And if you get a Y from dad, then your gender or your sex will be male.
Someone also asked for plants, what would you call the gametes? I assume they're also gametes?
Yeah, that's a really good one. Gametes as well. So gametes works on both, on any species that I can think of off the top of my head. If you wanted to be more specific, you could say the pollen and the ova in plants. You'd probably even get away with saying eggs for plants, but technically you should call it the ova. That's OVA. But yeah, definitely you'll get away with saying gametes. That's absolutely fine. It's a generic term. So as the sperm can carry either an X or a Y, there's a 50% chance that each fertilization will result in a male child, 50% of a female child. You get some really interesting genetic conditions where the gamete doesn't actually, the chromosomes don't separate like they normally would, and so you can get gametes that have like both X's in an egg or an X and a Y in a sperm cell, and then that can lead to individuals who have three sex chromosomes, and that leads to some quite unusual genetic disorders. That's moving a bit beyond the realm of level 1 genetics. All right, so what's the likelihood of John and Nolene's next child being a girl? This is a classic question that you want to be prepared for when you're dealing with Punnett squares in an exam. And so, as we've said already, there's a 50% chance of offspring being female. And what the question's really asking you here is for this next line. Each fertilization is an independent event. So every time a sperm fertilizes an egg, it's an independent event. That means it doesn't matter what's happened previously. Like two parents could have 8 children, they could all be boys, but every time there's another fertilization, it's a 50% chance of being a boy or a girl. So previous fertilization events do not influence the percentage chance of the gender of the offspring, the sex of the offspring. So we can wrap up our answer just by saying, therefore, the chance of their next child being a girl is still 50%. And that's true whether you're talking about sex determination in this case, or if you're looking at a Punnett square where you've got a more traditional monohybrid inheritance pattern, where you might be looking at the old 75 to 25% or a 3-to-1 ratio. So you'd be expected to be able to say that, each time fertilization happens, it's an independent event, which means that, any of the gametes could potentially form that offspring. And that one was a merit level response. Okay, so this question is dealing with lung disease, something called silicosis, and we're told that it's caused by breathing in stone dust. Now, straight away, looking at the question, you'll hopefully realize that breathing in stone dust is the influence of the environment. It's not the influence of genes. So it's not something that John has inherited from his parents. It's something that's happened to him throughout the course of his life, and it has no influence on his gametes and the chance of him passing that on to his offspring. If it was a genetic condition that was part of his that was in his chromosomes, then it could be passed on to his offspring. But this question is really just looking for us to identify, an environmental factor is not something that can be passed on to offspring. So in terms of the response, silicosis is caused by an environmental factor. It's not genetic and doesn't exist in the alleles that either John or Nolene will pass on to their offspring via their gametes. As such, it can't be inherited. The lung disease affects somatic cells, that's your body cells, not gametes. So when you're talking about gametes, that means sex cells, sperm, egg in humans. Somatic cells means your body cells, so that's every other type of cell that makes you up. And it's only the gametes that ever contribute genetic information to the next generation. I felt that the little bit in brackets there was a bit unfair. This was asked of you in the schedule, so it was identified in the schedule, the marking schedule, but it wasn't. I feel that you could answer the question without touching on that. So I've put it in brackets there just to suggest that sometimes you want to throw in that little bit more information, around the question without it necessarily being, directly in response to the question. So some extra information that it looks like the schedule was asking for last year. And I threw in here as well that the new baby could get silicosis if the new baby was exposed to stone dust, but the baby's not going to get that silicosis through inheritance. All right, and that was an excellence level response. Any other questions at the moment, Andrew?
Now we're all good.
Sweet. Okay, now with this one, I'm going to go into a little bit more detail about some of these terms. So we'll just look at the question, and then I'll pull in some other information for you. Got some images to share with you, and then we'll put together a perfect response. So here we're looking at a mutation in a human gene that can cause people to be protected from HIV infection. And we've got to explain the relationship between DNA, genes, alleles, and phenotype using this mutation as an example. So whenever you've got this sort of question, and really, any question that's giving you key terms that it's asking you to talk about, I would recommend ticking them off on the question sheet as you go through and do each one. And that's a really good way of making sure that you've covered absolutely everything. It's really easy sometimes to write out a beautiful answer and then think, right, sweet, move on to the next one, feeling that time pressure, but you might have missed one or two of the key terms that's going to make the difference between a grade for you. So use the opportunity to tick off, highlight responses so you can track that you have included those in your answer. Okay, so DNA, genes, alleles, and mutation. I'm probably going to throw a couple of other terms at us here because that's a classic question, and sometimes it'll throw in a couple of different words, like it might say, explain the link between DNA, alleles, genes, genotype, and traits, for example. So it might just change a couple of the words up, and I'll try and go through all of those terms that you might encounter here. So we've got a really nice image here of the DNA, the deoxyribonucleic acid, to give it its full name. And this is the chemical that forms our chromosomes. And we can see that it's made up of a series of what we call bases, nucleotide bases. And so these are the T's, G's, C's, and A's that you've hopefully encountered in your learning about genetics. And it's there's this sequence of letters that provides the code for every protein in our body. And a certain region on a chromosome provides the information for one protein, and we call that region on the chromosome a gene. Each chromosome might have, say, around about 20,000 genes on it. It's different depending on which chromosome you're looking at out of our 23 pairs. And so we've talked about the nucleotide bases, the chemical DNA, the chromosome. You can refer sometimes to this lovely shape here, this double helix. It's like that twisted rope ladder structure. And the fact that lots and lots of genes make up a single chromosome. So here we can see how that genetic material is coiled often very tightly. That helps it take up a lot less space and form these structures called chromosomes. You normally only actually see the chromosomes when the cell is about to go through division. So normally the nucleus just looks like a blob and you can't make out individual chromosomes. Now, when we've got those chromosomes, they're held inside the nucleus of the cell. So there's a few other terms that we need to include there. We've got genes, and there are, for any particular trait, there might be different versions of the gene. Humans are a really terrible example of this, but we often use them. The reason that we're not a very good example of this is that I don't think there's any cases of inheritance pattern in humans that actually follow monohybrid inheritance. So the examples that we deal with, we've kind of made them up to work in this context. But you've got to look at some really simple or much simpler life forms to see monohybrid inheritance really in existence. So when we've got genes throughout any human population, there'll be different versions of that gene. A crude example would be eye color. So you've got different eye colors. Each of those colors will have a slight difference in the sequence of bases in that region of the chromosome, in that gene. And so a different sequence of the bases means that it's going to produce a slightly different protein. The gene will code for a slightly different protein, and the slightly different protein will produce a different effect. It might be a trait that you don't see, such as perhaps the white blood vessel, the white blood cells that you produce. It might be a trait on those, so you're never going to physically see them, or it might be a characteristic that you can see on the outside of the organism. And so, there's your hair color, your eye color, your usual examples. So we call these different versions alleles. And in most examples, in all examples in level 1 genetics, you're only ever really looking at 2 alleles. One that we identify as dominant, i.e. that will always be expressed if it is present in the combination, and the recessive one, which is normally denoted by a lowercase letter, and that's the one that will only be expressed if it is expressed along with another recessive allele. So if you've got 2 of the recessive alleles, it'll be expressed. Otherwise, it will be masked by a dominant allele. So that's the genotype, the representation of the alleles, the letters that we use to show them is what we call the genotype. So the genotype represents the combination of alleles that an individual has. And then we talk about whether that's expressed or not, or which allele is expressed, and that's what we call the phenotype. So my eye color might be represented by 2 letters. It might be, I don't know, big B, little b, if I was heterozygous. And my phenotype is what would be visible. So that might be blue eyes, for example. Okay, so that's a whistle-stop tour of, we've done DNA, we've done chromosome, we've done gene, we've done allele, we've done genotype, we've done phenotype. I've touched on mutations. I'll just go back onto those a little bit more detail. So a mutation is a random, unpredictable, permanent change in the sequence of bases in a chromosome. So if we were looking at this one gene, and when the cell was copying, if a mistake was made and the sequence of letters or bases changes, then it produces a different protein, and that is a mutation. So that change in the DNA sequence is a mutation. Mutations happen all the time naturally. They will almost certainly have occurred in some of your cells throughout the course of your life. As you get older, you've been on the planet longer, you're more likely to have accumulated a few more mutations in your cells. Most of the time, they're so minor as to not have any effect, but occasionally they can be quite harmful. And if that causes the cell to duplicate rapidly and to grow in an uncontrolled manner, then that is what might make a cancerous tumor occur in the body. Mutations, the vast majority of mutations occur in your somatic cells, your body cells, so they're never going to be passed on to your offspring. But occasionally a mutation can happen in the gametes or in the cells that produce the gametes, and that means that the mutation can be passed on to offspring. Okay, so let's see how we can put that information together in an answer.
Just some questions about. So I think Michaela asked, where is the chromosome located in the DNA? She gets confused with genes. Also, is it the genes or the chromosomes, or both, that contain the base sequences which cause genetic mutation? And then the other question was, what's the difference between allele and phenotype?
Cool. Okay, I might need those questions again as I go through them because I'm not sure I'll remember them. I think the first one was asking about was it DNA or chromosome?
Where is the chromosome located in DNA?
Right. So the DNA is the chemical that makes up the chromosome. So the chromosome is this often drawn as an X-shaped structure, and the chemical that makes it is called the DNA. I'm trying to think of an example for you. Yes, you could say flour is the chemical, well, one of the chemicals that makes up bread. So bread might be the structure, but flour is the chemical that makes up the bread. And so that was a really quick off-the-cuff analogy. It probably breaks down at multiple levels, but hopefully that gives you the answer that you need. The DNA is the chemical. It's the name of the chemical that makes up the structure, this shaped object that is the chromosome. Next question was?
I think that's pretty much answered the first part. Difference between allele and phenotype.
Great. Okay, so an allele is a different version of a gene. So you've almost always got two alleles in your genetic information for any one trait. They could be the same or they could be different. And so those alleles literally just mean the versions of the gene. The phenotype is what is expressed as a result of the combination of those alleles. So we call the combination of those alleles your genotype, and then your genotype, and that's where the whole dominant recessive thing comes in, determines which of those alleles is expressed. And so the phenotype is we often say the physical representation of it on an organism. Whether they have the human examples are often like hitchhiker's thumb, a free hanging earlobe, or one that just kind of cuts straight into the side of the head, nose shape, eye color, hair color, etc. So those are the phenotypes. But remember, there are phenotypes that you're never going to be able to see. Like in this example, the mutation in the cell that makes you slightly more immune to HIV, you're never going to see that on the outside of the organism. But most of the time, the questions focus on phenotypes that will be visible. That was a really good one. Thank you. What was the third one, Andrew?
I think that was it.
Okay. If another one pops up, feel free to interrupt.
Sweet as.
Okay, this was an exam question I just thought we'd sideline on. So you've got to be able to pair up the nucleotide bases sometimes. So A's always pair with T's, G's always pair with C's. And there's the names of them there in full. That's adenine, thymine, cytosine, and guanine. But you don't often need to refer to them by name, just the letters. Okay, so back to our question. Let's see how we can fit this together in an answer. And hopefully seeing it written down will give you a chance to digest what I've been talking about. So deoxyribonucleic acid is the chemical which makes our chromosomes. These are the structures that can be inherited by offspring. Genes are distinct regions on the chromosome that provide code for a particular protein. One gene makes one protein, or provides the code to make one protein. Now, you can have different versions of the gene, and they're known as alleles. For most genes or traits, we have inherited two alleles, the only exception being where you've got that whole XY thing going on, and the X chromosome being longer and the Y chromosome being shorter means that males only have one allele for a lot of the stuff that's found on the sex chromosome. Sorry, men, that means that we're slightly genetically deficient in some ways. Just a simple fact of life. So for most genes or traits, we've got those two alleles, and that provides our genotype. So the combination of two letters is how we represent that genotype. Alleles can either be dominant or recessive. A dominant allele will always be expressed if present in the genotype. That is quite a good sentence to have prepared and ready to use in the exam. So, for example, those two letters should be, sorry, I've made a wee mistake there. So one of those genotypes should be capital T, okay? So both those genotypes would result in the same phenotype. That's the expression of a certain characteristic. A recessive allele will only be expressed if a dominant allele is not in the genotype. And so our example there would be little t, or homozygous recessive. homozygous, meaning both of the same type of allele, recessive, meaning they're both the non-dominant one. And a mutation is a random, unpredictable, I often include permanent change in the sequence of bases in DNA. And that will result in a different protein being produced, which may result in a different phenotype. But that's not going to enter the population as a whole unless it occurs in a gamete, and then that gamete goes on to be part of a new life, a new offspring. This change in the sequence can result in a new allele or version of the gene. In this case, a new allele may provide protection from HIV. So we've linked what's quite a generic answer, linking all of these different terms back to that idea of HIV, because we were asked to use this mutation as our example or in our explanation. Oh, and I should say, this is a nice picture of a human cell, and the HIV virus in this case are the little green dots, the little green blobs on the surface there. So this is a scanning electron micrograph image, I believe, that has been color rendered. So it normally just appears sort of gray, shades of gray, but color has been added to it afterwards. And that is an excellence level response.
Just one question on that. When we're talking about DNA and those types of questions, do we need to talk about its structure and how it's made out of sugar and phosphate bases / like the base pairing meters?
Not normally. I can't think of an occasion where you had to talk about or there might have been in the paper that had, where you had to draw this in, you might have had to talk about the sugar phosphate backbone of the DNA, but normally you don't need to go into detail about that. Probably one of those things that's good to know and be prepared to talk about in an exam if you have to. So, but you don't normally have to talk about the structure of the DNA any more than I already had. Good question. So discuss whether this gene mutation, which protects against the HIV virus, is helpful to the human population, and we should explain how it might be passed on to the next generation. So if the mutation occurs in the gametes, and this is really important here as to the human population, then the mutated chromosome could be passed on to offspring. If it occurred in a somatic cell, it can't be passed on and will not benefit the population. And in all likelihood, if it happened in one of your cells, it's not going to give you any benefit either. You know, the number of times that cell would have to divide to give you any real advantage is ridiculous. So, you know, the only real way that this mutation is going to give an advantage is if it's passed on to the offspring. Then in that offspring, every one of the cells will contain that mutated gene and may be able to produce a protein which gives some sort of advantage. So if the mutation is passed on, any offspring with this allele would be less susceptible to HIV. That means less likely to get HIV, giving them a slightly higher chance of surviving to reach reproductive age and passing on their genes. That sentence, you could almost take that from the giving them a slightly higher chance, blah, blah, blah. You could take that and put that into a lot of different questions of this type. So a lot of the time it's about identifying that some mutation gives an advantage, which increases the chance of surviving to reproductive age. And then if an organism reaches reproductive age, they've got a chance to pass on their genes, you know, carrying that mutated gene further and spreading it throughout the population. So over many generations, this slight advantage would raise the chance of the allele becoming spread throughout a larger human population, conferring protection against HIV to more of the human population. Now, there's a couple of really good examples of this, and I think I linked some little videos, maybe in where did I put them? I think I might have put them on that table with the survival and variation. You may have noticed a wee link on that where I was looking at the questions that exist. And if you click on that link, one of the links will take you to a Kurzgesagt video, which is a YouTube channel, and it talks about the gene that occurs in humans that allows us to digest lactose in animal milk. And large parts of the world don't have that gene and can't really digest large quantities of milk. But in Europe, that gene was spread throughout the human population, and so most Europeans can digest lactose from milk and can cope with drinking quite a lot of milk, whereas parts of the world, such as Asia, never really developed that gene because of different environmental pressures. I say developed that gene, sorry. The gene may have occurred, but it was never really reinforced through natural selection. So that's a really cool example you might want to watch and see if you can fit this explanation to that situation as well. And sorry, that was an excellence level response. Questions on that one, Andrew?
No, someone asked, do you need to structure the answers, or can you just write all the definitions and still get a good mark? And someone else mentioned.
Hold on, I'll answer the first question.
Yeah,
just so I don't have to remember them both.
Do
you have to structure your answer? You should structure your answer, and a good exam approach might be to jot down the key words and show how one is going to link to the other. So maybe just with arrows, doing a quick little plan that'll help you form your answer. So what I tend to do is I tend to think about the key words or key concepts I'm going to need to include in the answer. I might jot those down at the side. And then I would start my sentences, and ideally each sentence should sort of logically flow onto the next one. A series of definitions on their own would normally get you at least an achieved, assuming they were correct. But you might have to link the ideas together to unlock the merit and the excellence.
Cool. I think one of the replies was you need to relate it to the examples, and basically you have to relate it to the context of the question.
Yeah, and you can give a really good answer that doesn't relate to the question, but often there's just that little there might be a requirement in the schedule that you've got to link it back in, which is why you can see here the question talks about the HIV virus, and I've included HIV quite deliberately a couple of times in the answer. And in the previous example, the previous answer, I'd linked it in at the end.
Cool. I think that was it.
Awesome. Are we on question 3 already? Wow. Okay. So Himalayan wolves reproduce sexually. Explain how sexual reproduction causes genetic variation. And you should talk about the process of gamete formation, which is called meiosis, and fertilization. Remembering that those are two different things, but both of them can cause genetic variation. And meiosis happens first. That's the production of the sex cells, the gametes. And fertilization then happens when those individual sex cells, the sperm and the egg, fertilize to form the zygote. The zygote is the name of the single cell which is created after a sperm has fertilized an egg. All right. This is a really common question in exams. And so it doesn't matter, they might throw different examples at you. This year it's Himalayan wolves, another year it might be penguins, whatever it is, the answer's still looking really for the same information. And that's where that practice of previous exam papers will really help you, because you'll have a chance to again and again give a similar answer and refine it over multiple attempts. So to help us answer this one, I thought I'd drop down into meiosis. Now, this is probably more of a level 2 kind of diagram of what's going on. And I'm going to skip over a lot of the level 2 terminology because you don't need that. I'm going to highlight a few key things in here. So we've got something called independent assortment. So when the single cell starts that process of meiosis, the chromosomes align in what is called homologous pairs. Those are the pairs that are the same size. So out of your 23 pairs of chromosomes, one pair of them will be the same size, and then another pair of them will be a slightly different size. And so those homologous chromosomes are the corresponding chromosomes. And you'll have got one from dad and one from mum, and they'll pair up. So be able to use that term homologous chromosomes. So when those homologous chromosomes line up along the equator of the cell, we get this thing called independent assortment. And so what we can see in this diagram is that we've got the let's say that the blue one came from dad and the red one came from mum. In the first pairing of chromosomes, we've got the blue chromosome from dad, and that on diagram is on the right hand side. The red or pink chromosome from mum is on the left hand side. In the pair below, the chromosome from mum is on the right hand side and the chromosome from dad is on the left hand side. And so what's going to happen next is that they're going to get separated to opposite ends of the cell. Now, the example that we've got has only 2 pairs of chromosomes, but if this was happening in our cells, then there would be 23 pairs lining up down the equator of the cell. And with each pair, there's a chance that dads can go this way and mums can go this way, or it could be the other way around. And that means that each gamete is a random mixture of the genetic information that you got from your parents. So I hope that's reasonably clear, that idea of independent assortment and how it leads to how it creates variation. So it takes existing variation, the chromosomes and the genes on those chromosomes that were from your parents, and it mixes them up into a new order. Segregation is the name that we give to that pulling apart of those chromosomes. So independent assortment and segregation have to happen together for that variation, that mixing up of the existing variety in the genes that you have. In the same part of the process, we've got this thing called crossing over. And before we go on to look at that a little bit more closely, I'll just talk about this word haploid again. So the parent cell is diploid, which means it contains the full genetic component. In humans, that's 46 chromosomes in 23 pairs. And then after one cycle of meiosis, we get 4 genetically unique haploid cells. Genetically unique means that each one is genetically different, and haploid means that each one contains half of the genetic information that the original cell had. It's really important that each one has half the genetic information, because the male of the species has to produce a gamete, a sperm cell, with half the genetic information, and the female has to produce an egg with half the genetic information, so that the offspring has 2 halves, or the whole number of the whole complement of genetic information. If we didn't get that halving of the genetic information in the sex cells, then every generation would double the amount of genetic information, and very quickly there would be far too much genetic information in the cells. So that's a really important stage of reproduction. Now, crossing over, in the picture that you can see here, you'll notice that we start off with very distinct blue and red chromosomes, and then by this stage you can see a little bit of red has crept onto the blue chromosome, and over here a little bit of blue has crept onto the red chromosome. They've swapped over a little bit of each of their legs of chromatids, which are the sections, the legs, as I say, of the chromosomes. And in doing so, we've now got a chromosome that is genetically unique in that it's not the same as a chromosome that came from your father anymore, because it's had some of the genetic information from your mum's corresponding chromosome swapped over there. We'll have a look at that in a slightly bigger diagram. I think I've got next. So here's crossing over. The homologous chromosomes, remember that means that the chromosomes that correspond to one another, so they're normally about the same length, and each region on one of the chromosomes corresponds to a similar region on the next chromosome. So both of those regions would code for a particular trait, like eye color. Now, when they line up next to each other, they're really close, and the chromosomes can cross over, and the chromatids, that's the name for the legs of the chromosomes, can swap genetic information. So here we can see that the chromatids that have remained the same as they were at the beginning, so this red one that runs up here and this blue one that runs up here, they are non-recombinant. That means they haven't changed in any way. They haven't recombined. However, these two in the middle these recombinant chromatids, they have recombined by swapping over some of the genetic information. So the genes that were on this section from the mother, if you like, are now on the chromosome which was originally a duplicate of the father's chromosomes. I hope that helps.
If we ask some questions about the previous slides.
That's a good idea.
What's the first one? Difference between meiosis and mitosis. I think I sort of covered that. I just said meiosis was a production of the sex cells. Mitosis was a production of the rest of the cells, the somatic cells. Anything to add on that?
If you struggle to remember which one's which, mitosis occurs in mitose, and hopefully you know that your toes aren't part of sexual reproduction, and that would give you the clue that mitosis process is the cell division in other parts of your body. So if you can remember that, you know that meiosis is all about producing gametes.
Cool. Next question is, do you have to learn all the stages of meiosis?
Absolutely not. Absolutely not. And that was what I was saying. This is like a level 2 diagram. I've just put it in here because it highlights really well the 3 things that you do need to know about, which is independent assortment, segregation, and crossing over. The extent to which you need to know about them is really just that they are events that take place that introduce variation. You wouldn't necessarily need, you certainly don't need to go into the level of detail that I perhaps have in what's going on in those. The key things would be to know that independent assortment, rearranges the parental chromosomes, and crossing over mixes the genetic information that is on those chromosomes, creating genetically unique chromosomes, I suppose is how I put it. But you don't need to know the stages to the level of depth that I have described there.
Cool. That just answered someone else's question. There's another one, just wondering where segregation comes in within this, and if this happens at the same time as independent assortment. I'm pretty sure that just answered that.
I will go over it. I did mention it earlier, but it might have been a bit vague or unclear. So independent assortment is where the chromosomes line up down the equator of the cell, as we can see here. And then segregation is just the pulling apart, the separating of those chromosomes to opposite ends of the cell, which then influences which chromosomes are available to go through the next stage to make these haploid cells. So independent assortment and segregation have to happen together. The independent assortment lines them up, the segregation pulls them apart, but those 2 things combined, if one of them didn't happen, then you wouldn't get successful meiosis, you wouldn't get variation in the gametes.
And then the last question, which would be the best term between independent assortment and crossing over to talk about the stages of meiosis? Not quite sure what that means, but
both of them. You want to know both of them. So you want to understand that independent assortment, as I've said, rearranges the parental chromosomes. So instead of, in your in the sperm or the egg cell that your body produces having all 23 of your mum's chromosomes, it's going to have a random mix of your dad's and your mum's chromosomes. And that means that if you run the numbers, it's like 2 to the power of 23 from memory, which means that you're doing 2 times 2 times 2. Statistically, it produces something like 8.5 million different possible combinations of gametes. So that means that, just from independent assortment and segregation, you've got, 8,500,000. Sorry, start that again. 8.5 million different combinations. I haven't run those numbers on the calculator for a while, but hopefully that gives you a bit of an idea about the sheer scale of possibility there. Crossing over mixes that up further, which means that almost every gamete that is ever produced by your body is genetically unique. Might have gone off on a tangent there. Is that all right, Andrew, or do I need to elaborate?
I think that covered it. Someone asked, so do we relate independent assortment with crossing over, together with crossing over? I think they're 2 separate things, aren't they?
They are 2 separate things, but if you're going to be talking about them, you're probably talking about them in the same answer. So the question, like we saw with the Himalayan wolves, is about, what processes of gamete formation lead to genetic variation. And so both crossing over and independent assortment are important. So we'll see how I put that together in an answer in a moment. Just before we do the last little bit, fertilization. So remember, this is where the sperm fertilizes the egg to create a new organism, a genetically unique organism. There's a lot I'd love to talk about with this. There's some really cool science that can go on in here, but we'll keep it just for level 1 and just for your exam at the moment. And so here's the haploid nucleus of the sperm cell, the haploid nucleus of the egg cell, each with 23 single chromosomes. They recombine, gives you a full complement of 46. And that's what we call the diploid number. This picture shows you the moment that is happening. That's a sperm cell breaking into the membrane of the egg cell. And what will happen there is that the head of the sperm has a little place called an acrosome, which has some enzymes which allow it to breach the egg cell, the outside of the egg. And then the tail will drop off, and the nucleus, along with a couple of other bits, are inserted into the egg cell. And then the nucleus of both the sperm and the egg join together, giving the full complement. And it's amazing to think that out of about maybe 300 to 400 million sperm, every single one of you was the winning sperm. All right, and with that pleasant thought, we'll come to our answer for this one. So sexual reproduction is the creation of a new organism through random fertilization between a male and female gamete. During meiosis, independent assortment, segregation, and crossing over create genetically unique haploid gametes, each carrying a different mixture of parental alleles. Each gamete contains a random half of the genetic information of that parent, and these recombine in the zygote to give a mixture of the alleles. This mixture of alleles leads to different physical traits or phenotypes. I'm a little bit reluctant to use the word physical there because it kind of suggests that it's only physical. There's a lot going on inside you that you don't see that I'm talking about as well. And that is what we identify as genetic variation. So hopefully that answer clears up some of those questions for you about the level of detail you might need to go into. Often it's enough just to refer to those processes as being part of meiosis, but it would be good to know a little bit more, a bit like the independent assortment takes what's already there in terms of chromosomes and mixes them up, and then crossing over swaps over bits of the chromosomes to make genetically unique haploid gametes. I'm a little bit wary of time, so we might end up running slightly over. Here's the last question, I think. Himalayan wolves can live in places higher than 4,000 meters. They've got new combinations of genes that mean they can take more oxygen from the air. So explain the link between genetic variation and the survival of the Himalayan wolf population above 4,000 meters. This question really surprised me because we had that question about the HIV, which is effectively really talking about the same thing. How does genetic variation lead to improved chances of survival for a population? There's also a lot of really cool genetics and biology that I'd like to go into here, but I've tried to keep it just to level 1 for you guys at the moment. So above 4,000 meters, only the Himalayan wolves with a specific combination of genes are able to get enough oxygen from the air to survive. So that's me really just rewording a lot of what's in the question as a start to my answer. Himalayan wolves without these genes are, and here it is again, less likely to survive and reproduce. So there's that phrase that's really important to have prepared. And are less likely to pass on their genes. So the likelihood of survival, the chance of reproducing, and passing on their genes. That's a really key theme that comes through questions again and again and again. Since only those with the advantageous combination of genes can survive at this altitude, those individuals with those genes will interbreed and are more likely to pass on the favorable genes to their offspring. Over multiple generations, you're going to see the genes that confer that advantage being really reinforced in that population, so much so that they might become almost exclusive in that population, and you don't get a lot of the other varieties or the other alleles that aren't favorable disappear in the population through the death of any individuals that are carrying them. Okay. Interestingly here, I was listening to a talk by a guy called Steve Jones, who's a geneticist from the UK, and he talks about how humans climbing Everest, some people will climb Everest and use oxygen tanks, and some people will try and do it without oxygen tanks. Whether or not you can manage to climb Everest without an oxygen tank comes down to your genetics. If you don't have a certain combination of genes or a certain gene present on your chromosomes, then you are never going to be able to climb Everest without an oxygen tank. So it's fascinating that these examples are quite important in certain aspects of our life, even though we may never realize it. Yeah, that's an excellent answer there. Were there any other questions at the moment, Andrew? I was going to go through pedigree charts very quickly.
Yep. So someone asked, so you could potentially have two offspring that have the same genetics if it's like a 1 in an 8.5 million chance, but it's very unlikely. Yeah, it's next to impossible.
And it's more than a 1 in 8.5 million. And I'm not saying I've got that number right, but that's what I remember. Because that's only based on the independent assortment pulling a different set of chromosomes out that then create each haploid gamete. Every single pair of those is likely to undergo some extent of crossing over, and the crossing over can happen in different places along the chromosome, which means that you then take that 8.5 million chance and you blow it out the water. So much so that even though human males are producing 300 to 400 million sperm in the average ejaculation, and they're going to be recycling that sperm, that's any that's kept in the body anyway, repeatedly throughout the course of most of their adult lives. Even though you're dealing with such high numbers of sperm, it's incredibly unlikely that even any one of those sperm will be genetically identical. And then you start looking at the numbers and you go, well, okay, what's the chances of two of those sperm being genetically identical, in an ejaculation that goes on to fertilize an egg and produce an offspring, and you're just dealing with such astronomically small odds that it's never likely to happen. Where you can get genetically identical offspring is through twins, where one sperm fertilizes an egg, forms the zygote, and then during the stages of embryonic development, the zygote separates. Then each separate section of the zygote continues to form an individual. Then that's where you get identical twins, and so they are genetically identical. Yeah, hope that helps.
Someone else asked, do all 4 gametes from meiosis fuse into one cell, a sperm cell that fuses with the egg and fertilizes the egg? No, they stay separate.
Each of those 4 gametes is 4 different sperm.
Yeah.
So that gives you an idea how often meiosis has to happen to produce the number of sperm that we're talking about that are released from the body, and each of those individual sperm, only one will ever fertilize an egg.
Someone says, to get a higher grade, we're explaining the process of gamete formation. Do we need to go into depth, or can we just mention the term?
It depends on the question, I think. As I've said, I'd probably be prepared, if I were you, to talk a little bit about how independent assortment results in a mixing of the existing parental chromosomes, and crossing over creates genetically new chromosomes. That's probably the maximum extent I would go to.
Caroline asks, do we have to explain evolutionary biology when they ask about natural selection?
Yeah, so you need to be able to say that natural selection applies certain pressures to individuals, and those over time, those pressures, the selective pressures, favor certain alleles, and those alleles are the ones that, here's the phrase again, have an increased chance of survival, to reproductive age, to pass on their genes, to spread that allele further through the population.
Cool. And I think one more question. Can we just say that allele becomes dominant in the population because without the allele, those without the allele die off, and those that have the allele pass it on to the offspring?
That's a really good thought, but no, you wouldn't say that the allele becomes dominant. You might say that the allele becomes much more frequent in a population, or it might, that's a word I'm looking for. I want to avoid using the word predominant for the possible.
You could say it becomes more prominent.
More prominent, yeah. So its frequency in the population would increase. So you get a lot more of that allele if it was giving an advantage in a population over multiple generations. But if you then brought a member from another population in and they bred with those individuals with the successful allele, then any new gene might actually be dominant. So any new allele might be dominant and might overwrite the successful but recessive allele. So no, I wouldn't talk about it becoming dominant over time just because other versions no longer exist.
Cool, and that's it. That's all the chat.
Great questions, guys. Okay, we are over time a little bit, but I'll cover pedigree charts because this question didn't exist in 2021, but it's a really important one, and I would be ready to have a question on that in your exam. So this is about piebaldism, a genetic condition in horses that gives a white patch on the head and body. We're told that piebaldism is dominant, capital H, and normal color is recessive, lowercase h. Here's a pedigree chart, and we're told there's a little key at the side that tells us what the male and female piebald and normal are shown as. So list all the possible phenotypes and genotypes of horses 3, 8, and 9. So 3, 8, and 9. Well, straight away for the phenotype, we can see that 3 is piebald. And we can see that 8 is normal and 9 is again piebald, just using that key. So let's fill in that table. And then we come to the genotype. Individual 3 we're told they're piebald, and piebald is a dominant trait. However, they have had 3 normal offspring that, and in order to have those normal offspring, they must have passed on this recessive H. So in order to get these normal offspring, they must, 10, 11, 12 must both be homozygous recessive, little h, little h. Each of those little h's must have come from their parents, one from 3, one from 4. Therefore, we know that individual 3 must be heterozygous. They must have the dominant trait to be piebald, but they must also carry the recessive allele. Sorry, I should have said allele. The dominant allele, H, and the recessive allele, little h, in order to have the offspring that they have had and the phenotype that it has. So there's our heterozygous. 8 is normal, which can only be expressed if it's 2 recessive alleles, i.e. homozygous recessive, little h, little h. And number 9, so number 9 is piebald, which means they must have at least 1 dominant H. Now this is quite interesting here. They've had 4 offspring, and all of their offspring are piebald, which means that they must contain at least 1 capital H, at least 1 dominant allele. Number 8 that has mated with number 9, that's what the little line here means. Number 8 is homozygous recessive, little h, and so can only provide a little h to all of these individuals. Now, that means that all of these individuals must have inherited a capital H from number 9, but we don't know if number 9, based on that information, has 2 dominant alleles or is heterozygous, 1 dominant allele, capital H, and 1 recessive allele, little h. It looks likely that number 9 is probably homozygous dominant, but we can't be sure without, based on 4 offspring. Even if number 9 and 8 had, 1,000 offspring, then there's still a really small chance that the 1,001th offspring would inherit the recessive H from number 9 that previously had just never come through because each fertilization is a random event and it's a 50-50 chance. So the genotype for number 9 can be homozygous dominant or heterozygous. And that's quite a common question, asking you to discuss the likelihood of certain outcomes from a cross. Which brings us to a test cross. Now, a test cross, again, is a reasonably common question to see in the genetics paper. And a test cross is where you take an individual with an unknown genotype and you mate them with a homozygous recessive individual to look at the offspring and to try and figure out what the genotype of the unknown parent was. So in order for the parent to be unknown, it can't be homozygous recessive, because then it would demonstrate whatever phenotype was associated with that genotype. So it's only ever used in situations at level 1 genetics where you're trying to discern whether something is heterozygous or homozygous dominant. So to make sure the pair of horses were pure breeding, we could take them and pair them with the homozygous recessive individual. So if the breeder wants to produce only dominant piebald offspring, then they've got a breeding pair of horses, they've got piebald and normal horses. The piebald can either be heterozygous or, in the case that I'll show you next, homozygous dominant. And we'll be able to see from the offspring whether the individual that was used, the piebald individual that was used in part of that breeding, is what we call pure breeding, homozygous dominant, meaning they'll only ever pass on that dominant allele, or heterozygous, which means they've got a 50-50 chance of passing on either the dominant or the recessive allele. So here are the gametes that can be formed from each of these parents. And this is the Punnett square filled in. So we see in this case that there is a 50-50 chance of the offspring being piebald or being normal. Now, if you produce multiple offspring with this pairing, then hopefully at some stage you would see that either one was normal, and in which case you would know that the adult, the parent here, had to be heterozygous, so not pure breeding. Let's contrast that with another example. So again, test cross, we're using homozygous recessive as our test cross, and we've got homozygous dominant as our what the piebald horse could have been. So previously we looked at heterozygous. Now we're considering what would happen if it was homozygous dominant. Those are the gametes that can be formed, and that means that all of the offspring have both the dominant and recessive allele. So all of the offspring, 100% of them, are going to be piebald in this example. And if we have enough offspring from this pairing, then we could say with reasonable certainty that this individual was pure breeding. You can never be 100% sure, because there's always the chance that it was actually heterozygous, and just by fluke, you've had only this allele ever being passed on to those offspring, so the dominant allele being passed on to offspring. So you can never be 100% sure, but the more offspring that are produced from that pairing that are normal phenotype, piebald phenotype in this example, the greater your certainty that this adult, this parent, was pure breeding or homozygous dominant. All right, and that brings us to the end of the genetics webinar for this year. Are there any last questions?
No, that looks to be it.
Awesome. Oh, well, thanks very much, guys. Good luck for your exam. Use those resources to help you, and I'm hoping and I'm sure that you will, if you put the effort in, then you will succeed. All the best.